In this video, we will discuss about some of the basics of phagocytosis. So, in the phagocytosis, what will happen is that the any any foreign or harmful particles that is been detected by the immune system will be eaten up by the phago, phagocytosome, and this process is known as phagocytosis. So, how this take place in general is that there are several receptors that are available on the phagocytosome, and some of these receptors are shown here that take place in this kind of antigen detection. There are other receptors are also available, such as Fc gamma R1, Fc gamma R2, Fc gamma R3, and also these complement receptors such as CR3 and other complement receptors are also there. So we will understand the phagocytosis from one of the examples that is shown in this figure. So in this case, this is the antigen that has been detected by the phagocytosome, and it has been detected by this Fc gamma R2a, and as soon as the Fc gamma R2a interacts with this antigen, it will trigger the downstream phosphorylation or downstream signaling pathway because of the phosphorylation of this Fc gamma R2a that leads to this RAP-GTP activation, and this RAP-GTP activation leads to the activation of this integrin C3, that it opens up from this closed positions to this position, and it will also help in binding the antigens to this phagocytosome, and simultaneously it will keep off the other high receptors such as CD40 5R, so that uh, engulfment of these antigens by this phagocytosome can be happen smoothly. And so this is what happens when the antigens interact with these receptors on the phagocytosome. And this is just a brief, and there are several other pathways that get activated, and in some of the details are shown in this top middle figure. In this case, we can see that when this it is a different example. In this case, when this Fc gamma R2a gets activated, then it will lead to the phosphorylation. It is just extended example of this first one, not a different example. So once it gets activated, it will lead to this complex formation of this LAT that lead to the activation of PLC gamma, and this will activates this IP3, and that will lead it will activate IP3 and also this PKC and extracellular signaling pathways such as this ERK and P38. It will take care of that, or that will dysregulate this calcium within this phagocytosome. And simultaneously, on the other hand, this SYK activates this VAP that will lead to the activation of this RAC. RAC will also be activated by this PIP2 through PIP3, and this PIP3 will leads to this myosin activations and simultaneously this RAC activations, and this will cause this polymerization of the actin, and this GNK and NF kappa beta pathway will also gets activated. So these are some of the pathways get activated through this Fc gamma R2a, and from this integrin CR3 will also leads to activation of some pathways that is shown in this top right figure. So in this case, when this integrins gets interact with the antigen, this lead to this Rho pathway activations, and this will also lead to this myosin 2 that will lead to the myosin 2 activations, which leads to this ARP23 complex formation and the actin polymerization. And Rho can directly activate this actin polymerization through this MDA, and this will also cause more macrotubule formation in the cytoplasm. So these are the some of the pathways that get activated when the within the phagocytosome when it interacts with the antigens, foreign antigens or harmful antigens. And this is just a brief overview. So when the antigens gets interacted, how these phagocytosomes try to engulf these antigens. So it will engulf the bacteria or the foreign harmful particles that has been detected by the immune system, and it will engulf in three different stages. First one is the early endosomes. In this case, the vesicles that has been used for engulfing the bacteria will be recycled, and at the same time some of the some of the bacterials or foreign or harmful particles components gets degenerated, and after that it will go into the late lysosome where this bacteria where the further part of this bacteria will get degenerated. At the same time, the vesicles which can recycle is recycled back into the membrane of the phagocytosome, and thereafter it will go into this final step, that is lysosome or phagolysosomes, where all the bacterial components will be degenerated and all the amino acids can be recycled back within this phagolysosome, within this phagosome. uh, for this phagosome cell cycle process. This is how this phagocytosis occurs within the cells. So this is just an example. There are other several things might have occurred during this phagocytosis that are not covered in this video. But the purpose of this video is to get you a basic idea about phagocytosis and with the help of an example. If you need to understand more about this phagocytosis, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.